Hello, and thank you for the introduction. And today, we will be speaking about pulling back the curtain on the next generation of human milk oligosaccharides, really focused on new findings from preclinical research. During this presentation, we will discuss the diversity of human milk oligosaccharides in mother's milk. We will also explore some new findings in preclinical research to really understand the mechanisms by which we may be able to explain some of those unique benefits that we see. Benefits which have been well published in the clinical literature. These include things like immune health, gut health, brain health. But we'll also explore some new avenues of potential future benefits which are seen currently in preclinical models. What we all know from years of evidence and centuries of human evolution in anthropology biology is that improved rates of exclusive breastfeeding led to reduced rates of respiratory and gastrointestinal infections, reduction in overweight and diabetes later in life, probable protection from certain atopic conditions, and a benefit of improved cognitive development. The multiple and diverse group of HMOs can be really classified generally into three main categories, largely based on their structural building blocks. They're the non-fucosylated or core human milk oligosaccharides that are neutral in nature. These HMOs are the foundations upon which other HMOs are built. LNT is the most abundant representative in this category. Another HMO is the fucosylated HMOs category, which are also neutral in nature. 2FL is the most abundant fucosylated HMO, and DFL, also known as LDFT, is among the 10 most abundant representatives in this group. Sialylated HMOs are acidic in nature, and this is the third category. Amongst them, 6SL is the leading representative, and 3SL represents an important, even though smaller, sialylated HMO. The current evidence suggests that the structure of an HMO determines its function. So much like we see in other areas of biology, not all things are the same, and in this case, not all HMOs do the same thing, so the diversity is important. You can see in this framework here that we will really talk about a few of the established areas of impact, like immune health, gut health, and the impact on neurocognitive development. But we will also explore the mechanisms behind some of these benefits, including the impact of HMOs on the microbiome, their impact on epithelial barrier function, and immune and cellular pathways that they directly or indirectly impact. So let's start with mechanisms that may explain some of the clinical findings related to immune health. Experimental and preclinical studies have provided evidence that certain individual HMOs can decrease the load of pathogens that are associated with increased respiratory or inducing respiratory infections. 2FL or 3SL decrease the pathogen load of respiratory syncytial virus, and 3SL or 6SL decrease the load of Pseudomonas aeruginosa, really pointing to the structure specificity by which these HMOs act. Furthermore, the single HMOs of 2FL, 3SL, or 6SL can also impact rotavirus infectivity in vitro, as well as the combination of 3 and 6SL. More recently, 2FL reduces Campylobacter jejuni infectivity in vitro and in animal models. In addition, 2FL decreases adherent invasive E. coli infections and reduces intestinal inflammation in some preclinical animal models as well. Let's take a deeper dive. To uncover the novel physiological functions and pathways of HMOs, Fuata and colleagues screened a panel of over 100 G-protein coupled receptors, GPCRs, using a blend of 6 HMOs. These include 3SL, 6SL, LNT, LNNT, 2FL, and DiFL. and they followed up on the positive hits with standard receptor assays. What they showed us was LNT and 6SL individually activated GPR35, and they showed synergy when used together. In addition, in vitro fermentation of infant stool samples showed that 2FL can upregulate the production of GPR35 agonist kynuric acid by the microbiota. So what does this mean? This tells us that activation by 6SL and LNT on GPR35, which is really a receptor that mediates attenuation of pain and colitis. And this can have an impact, but needs to be studied clinically. One of the well-studied animal models of inflammation, which we all know about very well, is NEC, or necrotizing enterocolitis. Breast milk is rich in non-digestible oligosaccharides and prevents NEC through mechanisms which we don't completely understand today. In this study, NEC was induced in newborn mice and premature piglets. What they found was that supplementation with either 2FL and or 6SL, but not the parent sugar lactose in isolation, reduced NEC in mice and piglets via reduced apoptosis and reduced inflammatory pathways. Mechanistically, both 2FL and 6SL, but not lactose, reduced toll-like receptor 4 mediated inflammatory signaling in the mouse intestine. In a really good article that was published by Zhao et al., and it was published in Frontiers back in 2017, they aimed to determine the effect of 2FL on vaccination responsiveness, both innate and adaptive, in a murine influenza vaccination model. And they wanted to elucidate the mechanisms that were involved. They found that 2FL significantly enhanced the vaccine-specific DTH, which was the delayed type hypersensitivity responses, accompanied by increased serum levels of vaccine-specific immunoglobulins, in this case IgG1 and IgG2A, in a dose-dependent manner. They concluded that dietary interventions with 2FL improves both humoral and cellular immune responses to vaccination in mice, which might be attributed in part to the direct effects of 2FL on immune cell differentiation. So now let's talk about the role of HMOs impacting through the microbiome. What we know from data that is published to date is that HMO usage by infant gut microbiota seems strain-specific and HMO structure-specific. As shown in this study here by Florence Rochat and Séverine Combremont, which presented in June last year, they found and examined the in vitro ability of various bacterial strains, either isolated from infant stool or used as probiotics for HMOs, and looked at their HMO usage for growth and metabolic activity. What they found was that probiotic and commensal bacteria, when incubated with HMOs 2FL, diFL, LNT, LNNT, 3SL, and 6SL, or phosphogos for 24 to 48 hours in anaerobic conditions, with lactose and glucose used as controls. Their findings teach us, and what is really interesting, is that 2 strains of Bifidobacteria bifidum were able to grow on all individual HMOs and their combinations. Only 2 B. longum strains grew on 2FL, and 1 on LNNT. However, certain other strains of staph did not grow, showing the selectivity. Hence, they concluded that HMO use is highly strain-specific and HMO structure-specific. LNT and LNNT and 2FL were among the most used HMOs when looking at sole carbon source usage. In another study presented at the conference last year, Norbert Sprenger and the group examined the impact of HMO or their combinations on toddler gut microbiota in a preclinical model. What they showed us in this preclinical model is that there was promotion of actinobacteria and a decrease in the relative abundance of Firmicutes and Bacteroidetes. In the actinobacteria group, mainly Bifidobacterium, and specifically Bifidobacterium pseudocanthulatum, increased. They also demonstrated the fermentation of HMOs strongly increased the production of lactate and acetate. These data provide the evidence that HMOs do impact toddler gut microbial growth and metabolic activities in a preclinical model, and really shows us the impact of these HMOs on shifting their metabolism from a proteolytic to a saccharolytic metabolism. The short-chain fatty acids formed are potentially reasons that we may hypothesize contribute to gut health benefit effects. In an elegant study done by Anne Lin and Lars Bode's group, which was published in 2017, they uncovered a novel HMO property to directly inhibit the growth of group B strep, independent of host immunity. By separating different HMO fractions through multidimensional chromatography, they found the bacteriostatic activity to be confined to specific non-sialylated HMOs and synergistic with a number of conventional antibiotic agents. They identified a mutation within a GBS-specific gene that confers resistance to HMOs, suggesting that HMOs may function as an alternative substrate to modify a GBS component in a manner that can impair their growth kinetics. Their study uncovers a unique antibacterial role for HMOs against the leading neonatal pathogen and expands the potential therapeutic utility of these very versatile molecules. Further experimental preclinical evidence support the role of HMO diversity on gut health. 6SL and 3SL, single and in combination, increase the adhesion of beneficial gut bacteria to human epithelial cells in vitro. The reduction of visceral pain induced by chronic stress was shown for a combination of 2FL and diFL, as well as LNT and 6SL in an animal model. Furthermore, 2FL seems to regulate gut contractility in vitro, and there is new evidence that each of the HMOs, 2FL and LNT in particular, strengthen gut barrier function in vitro. So let's look at some of this visceral hypersensitivity data in a little bit more detail. In a mouse model of visceral hypersensitivity, we saw additional structure-specific impacts of HMOs, in this case, particularly 2FL and diFL, in reducing gut sensitivity to distension in the mouse model of visceral hypersensitivity. While all the HMO combinations showed a directional benefit, it was particularly pronounced with 2FL and diFL. Multiple studies from various groups, particularly the work done by the University of Illinois, has taught us that specific HMOs in ex vivo and animal models can have benefits. In their data, increased brain sialation, attenuates stressor-induced anxiety, increased spatial recognition, memory, as well as attention, and also showed effects on the affecting hippocampal functioning, like learning and motivation. When we talk about newer avenues, among the first one where benefits have been looked at, particularly early life HMO exposure, was on later bone health. As you can see from this data here, that HMOs can really have an impact on ensuring bone quality, as you can see from the specific data of the impacts of either 2, 4, or 6 HMOs on bone structure. However, these findings need caution as they need to be validated clinically. The immunomodulatory effects of HMOs have really taught us that HMOs may have some therapeutic potential in allergy. Castillo-Cortate and colleagues investigated the effect of HMOs 2FL and 6SL on food allergy symptoms in a mouse model. Anaphylactic symptoms were induced by oral ovalbumin, and these were done in challenge in sensitized mice. They were given a daily oral treatment with either 2FL or 6SL or lactose. Treatment with either 2FL or 6SL suppressed mast cells in the intestine, TNF reduction, passive cutaneous anaphylaxis, and resulted in significant attenuation of oral ovalbumin-induced intestinal allergy as assessed by the diarrhea score, as you can see from the graph here. Daily oral treatment with 2-FL significantly reduced food allergy symptoms such as diarrhea and hypothermia in this animal model. The authors suggest that certain HMOs may have a direct therapeutic potential in allergic disease, which hopefully will soon be clinically explored. So in conclusion, we can confidently say that diversity of HMOs is important, and the structure of HMOs influence their structure-specific function. To summarize what we've learned today, 2-FL, LDFT or di-FL, LNT help to shape the early gut microbiome maturation. 2-FL, LNT, 3-SL, and 6-SL help immune competence development and explain mechanisms mainly related to reduced infections, but as we can see from the last data, even potential opportunity in their mechanisms for allergies. 2-FL, 3-SL, 6-SL support central and enteric nervous system development, as seen in the gut contractility, and also in some of the data on their cognition benefits. And newer benefits in bone health and their influence, particularly as it relates to the treatment of certain allergic outcomes, continues to be explored. Thank you.